So we are very thankful for Aaron Cechanover that he has shown us again that basic understanding opens the door to clinical application. And that's somehow in the core of this conference. We would like to move from a so-called evidence-based medicine where we look at big statistics of patient cohorts and then we believe we understand what happens, to a knowledge-based medicine where we understand what happens in an individual patient and then act on this base. And that's why it's very apt that the next speaker is Professor Andy Clark from Birmingham, a specialist in inflammation, because inflammation is another topic which really sits at the heart of so many diseases, and we still have so little understanding of it. So please.
Good morning. I'd first of all like to thank the organizers, in particular Bayat, for the invitation to speak here. I'm very unlucky to have to follow a talk as good as that one, but in some other respects I'm quite fortunate. I'm fortunate with the title that I've been given for my talk, because resolution is exactly what I want to discuss with you, the importance of resolution and absence of resolution in chronic inflammatory disease. And I'm also fortunate to be working in the field of inflammation biology, because inflammation touches everything. It's not only a defining characteristic of all of these pathologies with itis in their name, it's also a causative factor in many other important pathologies as listed here. Through many years of research, we've come to understand a lot about how inflammation is initiated. And to a large extent, that's a property of pattern recognition receptors. These are receptors that have evolved in order to detect the presence of pathogens. And they do this by recognizing and sensing small molecules or classes of molecules that are associated with particular pathogens. And we have several families of pattern recognition receptors. Not all of them are shown here. And it's important to remember that several of these pattern recognition receptors also recognize molecules that are produced at sites of tissue damage, damage-associated molecular patterns. For example, proteolytic fragments generated by the cleavage of extracellular matrix proteins. And that's an important property which allows the innate immune system to be activated, inflammation to be commenced in response to tissue damage even before a pathogen arrives. Decades of research have taught us a lot about how these signaling pathways function. An example here is the toll-like receptor 4, which is a receptor for lipopolysaccharide, a molecule that's associated with the cell wall of gram-negative bacteria. And when it detects LPS, this receptor fires off a signaling cascade, part of which is shown here. And this includes ubiquitination events. These are lysine 63-linked ubiquitinations, and also there are several phosphorylations that occur. And one of the end results of this signaling cascade is the activation of the transcription factor NF-kappa B. And NF-kappa B induces the expression of inflammatory mediators, for example, the cytokines TNF and interleukin-1. And TNF and IL-1 themselves are, of course, pro-inflammatory molecules. They initiate their own signaling cascades, which resemble this one, and they lead to the activation of NF-kappa B as well. So these signaling pathways have a number of very important properties. The first is extreme sensitivity. The second is rapidity. They're able to activate transcription of target genes within a matter of only a few minutes. These signaling pathways are largely based on protein-protein interaction, and of course these interactions can occur just by chance within the cell, so these biological systems always contain noise. And there's a property of amplification. The amplification arises in a number of different ways. Firstly, several of the steps of the signaling cascade are enzymatic, catalytic, and that conveys amplification of the signal from the cell surface into the nucleus. And also, because the end products of the signaling cascade are themselves pro-inflammatory, they cause recruitment and activation of other cells, they cause tissue damage, and therefore they generate novel ligands for the pattern recognition receptors, and this gives rise to amplification. And these properties are absolutely essential for our survival in a world that is filled with pathogens. But obviously, they also create an enormous danger. and that is the danger of unprovoked, uncontrolled, runaway inflammatory responses. And of course, in most of us, most of the time, that doesn't happen, because no biological system works in this way. There are always balancing negative feedback mechanisms in order to restrain an inflammatory response and to promote resolution of inflammation. And I'll show a couple of examples of that here. As well as activating the expression of TNF and IL-1, NF-kappa B drives the expression of I-kappa B alpha and A20. A20 is also known as TNF-AIP3. And these are what I call negative feedback genes. And their function is to feedback and inhibit the NF-kappa B signaling cascade in order to terminate that inflammatory response and to make sure that the inflammatory response is appropriately resolved in a timely fashion. If we just consider macrophages, this is my favorite cell type, we discover that the feedback loops operate at a number of different levels. Firstly, there are intracellular feedback loops like the one I've described to you. Secondly, there are local or paracrine negative feedback loops. For example, activated macrophages produce soluble cytokines like interleukin-10 and TGF-beta, which are anti-inflammatory and feedback in a local fashion to switch off macrophage activation and to prevent further expression of inflammatory mediators. And there are also systemic feedback loops. For example, the existence of pro-inflammatory cytokines in the circulation is sensed by the hypothalamus. This activates the hypothalamic pituitary adrenal axis, the HPA axis, and this leads to the production of glucocorticoids, principally cortisol, from the adrenal glands. And cortisol, of course, is an extremely potent anti-inflammatory hormone which inactivates the macrophage and again promotes resolution of this inflammatory response. So how do we know that these feedback mechanisms are so important? There are several different lines of evidence. The first comes from genome-wide association studies, and this is just one example. This may not project too well, but this is a study on TNF-AIP3. This is the feedback gene that's partly responsible for switching off NF-kappa B signaling. And polymorphisms within the locus for TNF-AIP3 are found to predispose to very many chronic inflammatory diseases and autoimmune conditions. That's just one example. The second line of evidence comes from genetically modified mice. And again, this is just one example. This is a tissue-specific knockout of the TNF-AIP3 gene, and the consequences for the mouse are rather dire. The mouse develops an erosive arthritic disease, which is very similar to rheumatoid arthritis. And experimentally, it's also possible to improve the efficiency of these negative feedback mechanisms. And this is an example from our own work. We're studying a negative feedback gene, which is called ZFP36. We've engineered 2-point mutations within that gene, altering phosphorylation sites. It's a gain-of-function mutation so that in this mouse, the negative feedback control of inflammation works more efficiently than it does in a wild-type mouse. And this mouse is extremely resistant to inflammatory insults. In this particular experiment, we've induced arthritis in wild-type mice. In the genetically modified mice, we cannot induce arthritis. They also don't develop thrombosis. We can't give them sepsis. And in some preliminary experiments, we find that they're also resistant to tumor development. So the system can be made to work more efficiently. And the final line of evidence comes from successful anti-inflammatory drugs which are used in the treatment of these chronic diseases, and they are very commonly found to switch on the expression of negative feedback genes, and to some extent this is how they work. This is certainly the case of glucocorticoids. They switch on the expression of a number of negative feedback genes and help to resolve inflammation in that way. This begs a very obvious question. If there are so many of these negative feedback mechanisms, they're so powerful and redundant and sophisticated, how on earth do we ever arrive at a state of chronic inflammation? And to me, the answer is obvious. This is a little bit of a statement of personal belief. In order to get to a state of chronic inflammation, there must be some failure of these endogenous negative feedback or pro-resolution mechanisms. I think the important question is, To what extent is that failure of negative feedback a causative event in the development of chronic inflammation? If I can use a metaphor that has been used already once this morning, if you discover a car crash at the bottom of a steep hill, you may be wasting your time by asking, why is there a hill? It's not difficult to start inflammation. It happens all of the time. Perhaps it would be more useful for us to ask, is there a problem with the brakes? I work in a rheumatology research group. My colleagues are clinical rheumatologists. I'm not myself a clinician. This is how they describe the natural history of rheumatoid arthritis. There is a genetic predisposition to RA, which increases the probability that you will develop this disease. At some stage, there may be a pre-symptomatic phase where autoantibodies are detectable, but there are not yet any symptoms. And then there's a gradual progression through these stages of the disease to a final stage, which is erosive, destructive, painful, debilitating, and accompanied by intense collateral damage to other organ systems, such as the cardiovascular system. Now, patients are not convenient animals. They tend to hop around from one phase to the other, rather than progressing neatly from A to B to C to D, etc. But more importantly than that, they can also move in the other direction. So some patients who present in clinic with apparently severe rheumatoid arthritis can spontaneously resolve their joint disease without having been treated. And these patients always tend to disappear from sight, which is an intense tragedy, because I feel that we can learn an enormous amount from these patients who have a natural ability to resolve inflammatory disease. There's an increasing pressure to treat patients early, not only for rheumatoid arthritis, but for many other chronic inflammatory conditions. And this graph illustrates exactly why. This shows clinical outcomes in groups of patients who've been treated early, within 3 months of the onset of symptoms, and their disease progression is relatively benign. There's a slow increase of clinical score. But if there is a delay before the commencement of treatment, doesn't matter which treatment, the progression is steeper, the outcomes are worse. So this generates a great deal of pressure to try and identify patients as early as possible and to begin aggressive treatment. But here's the problem. You will end up treating patients who would automatically resolve their disease without any treatment in any case. These drugs are not only expensive, they obviously have side effects. So one risks causing harm to patients who are not in need of aggressive treatment. So these patients represent not only a challenge, but also an opportunity to learn about how these pathways to progression and to resolution are handled and how those decisions are made. My clinical colleagues have established a cohort which they call Beacon. They've made an extreme effort to capture patients with very early symptoms of joint inflammation. That's defined as a duration of symptoms less than 3 months. And their recruitment to the cohort is not dependent on diagnostic criteria, it's just on symptoms of joint inflammation. And within this cohort, we have about 50% of patients who spontaneously resolve their joint inflammation. And there is currently no basis for predicting which way they're going to go. However, when we examine their serum and we carry out metabolomic analysis, we find that we can actually separate them. These are patients with early arthritis. We can't separate them on the basis of the severity of inflammation, serology, or any other criteria, but on the basis of their metabolic profiles, we can tell which ones are going to spontaneously resolve their disease and which ones are not. And this is my final slide. I think the major challenge in chronic inflammation, this is a personal view, is how we can quickly identify those patients who need early and aggressive treatment of their symptoms. But more importantly than that, how best can we learn from those patients who do not need that treatment, who are going to resolve their disease in any case. And the rest of this I'll just leave on the slide because I'm slightly over time. Thank you very much.
Thank you very much for this enlightening presentation. We have the discussion after the 3